To view plant cells, the plant we use is an onion. So we chop up an onion into small pieces, and taking a small piece of onion, we can either use a forceps, if necessary, or fingernails, and you remove a piece of onion epidermis from the inner side of the curve of the onion and place it onto a microscope slide. Again, to prevent dehydration, we add a few drops of water. Again, we need a cover slip, and we place the cover slip, the edge of the cover slip, down into the water and lower it slowly using the needle to remove air bubbles. We place the slide onto the microscope. Again, lower the stage of the microscope. Place it on. Always starting with the low power lens, so click the low power lens into position. Bring the stage up to its maximum. This time I might put on the clips. And viewing it, we rotate the microscope, the stage slightly downwards, and we see a clear image. Again, to adjust the light, we just rotate the diaphragm and you select the light that you find most suitable. To view the cells under medium power, just rotate in the medium power lens. And again, a slight refocus. And to view under high power, rotating in the high power lens, and this time using the fine focus. Slight rotation gives us a clear image. To view plant cells stained, again we use a piece of onion. You can use the forceps or fingernails if suitable. And we peel off a piece of onion from the inner curve of a piece of cut up onion. So a very thin layer. This allows light to pass through. Place it onto the microscope slide. As a stain for plant cells, we use iodine. So 3 drops of iodine solution, it being a kind of a red, yellow colour. Again, we add a cover slip and we add it at an angle. So the cover slip goes down into the solution. Using a needle, we lower the cover slip at an angle to remove air bubbles. Slowly removing it. We now transfer the slide onto the microscope stage, lowering the microscope stage to allow us to work clearly. And we center it, clip it into position. Again, bring the microscope, click in the low power lens, starting with low power. Bring the microscope stage up as high as possible. View it under the microscope, and using the coarse focus, turning it slightly, we adjust until we see a clear image. Again, the diaphragm can be used to change the light intensity and produce a clearer image, hopefully. To view under medium power, we click in the medium power lens and use the coarse focus. And to view under high power, we use the high power lens, and this time we use the fine focus. Images of the unstained and stained onion cells.
The aquatic plant Elodea is a great introduction to the microscopic world of plant cells. Let me show you. So Elodia is the easiest of the wet mounts to make. So you want to get your slide ready and waiting. Put a drop of water on the slide. You want to select a leaf from the top of the Elodia plant. And place that in the same orientation on your slide. So the upper surface of the leaf is upward on the slide. Add your cover slip, place it at an angle and drop it down over your specimen. And if you need to add additional water, you can add a drop at the corner of your slide. And that's it.
To view animal cells, the source of the animal cells will be cheek cells. To obtain the cheek cells, we scrape the inside of our cheek. You can use a cotton wool bud, but sometimes that produces small strands of cotton wool or cellulose. So in this case, we'll use a wooden spatula. And we'll scrape the inside of the cheek using a wooden spatula. We now transfer the cells, saliva containing the cells, onto a microscope slide, and we just center it. To prevent the cells from dehydrating, we add a few drops of water. Normally about 3 drops of water would be sufficient. We now want to add a cover slip. The water could wet some of the lenses. So we add a cover slip and we place the end of the cover slip into the water there. And to eliminate air bubbles, we use a needle or something similar to lower the cover slip slowly, very slowly, and I withdraw the needle very slowly, and the water runs along the slide and pushes out and eliminates the air bubbles. We now transfer the slide to the microscope. Again, rotating the coarse focus so that this microscope stage is at its lowest point. Clicking in the low power lens, and we center it. So we bring the stage up as far as it will go. And then looking in, we focus. We may have to center the slide sometimes just to get the cells right, and we'll clip them into position. And focus. We can then see the cells at low power. To adjust the light, we can adjust the diaphragm, and that may give us a clearer image, or not, as the case might be. So we keep doing that till we find the image or the light intensity that suits best. To observe the slides then at medium power, we rotate in the medium power lens. Usually very little adjustment of the coarse focus here will do the job, and that brings it into focus. And finally, for high power, rotate in the high power lens, and this time using the fine focus, we can produce or see a clear image. To view stained animal cells, again we use cheek cells. Using the wooden spatula, we scrape the inside of the cheek to obtain the cells. We transfer the cells onto a microscope slide and again center them as before. This time, instead of water, we're going to use methylene blue. Methylene blue is the stain that's used to show animal cells. It highlights certain parts more clearly. Usually about 3 drops of methylene blue would be sufficient. Again, methylene blue would be damaging if it came in contact with the lenses. So to protect the lenses, we add a cover slip. Again, we touch the base of the cover slip down into the methylene blue and then gently lower the cover slip so that the air is pushed out and there are no air bubbles in the final slide. I'll just tidy up the slide a little bit. Transfer the slide onto the microscope stage, moving it down. Again, rotating the low power lens. Looking down, bring the microscope stage up to its highest point. And always you're focusing downwards, so that means that the slide doesn't get broken or damaged by the lenses. If you were focusing upward, the slide might collide with the lens and crack and break, etc. So we're focusing in downward direction, and a little gentle focus, and the cells can be seen. We can then switch to medium power. Just place the clips in position first. We can switch to medium power and to high power, and adjust or focus accordingly. All the time remembering to change the diaphragm and pick out the light intensity that's most suitable. The results of the unstained and stained cheek cells can be seen on the slide.